Welcome to today's video session. In today's video, we are going to discuss about photorespiration, which is a very important, significant phenomenon occurring in green plants. So, before moving on to this video, I request you all to watch my introductory video on respiration and also Calvin cycle, because when those concepts are clear, this will be much more easier and clearer for you. So, for those who didn't have watched those video, please I request you all to watch those video. So moving on to this photorespiration, it was discovered in the year 1955 to 1963 by two eminent scientists called as Decker and Krotkov. And later it was also discovered by Hatch and Osmond in the year 1976. They discovered where actually this place takes place. They found out that this was occurring in three cell organelles, namely chloroplast, peroxisomes, and mitochondria. So chloroplast, as we all know, it is a type of plastid where chlorophyll pigments are present that is responsible for photosynthesis. Peroxisome is also lipid bilayer membrane. It is like a mitochondrial membrane only. It is also like say other cell organelle where the photorespiration is taking place. And mitochondria, as we all know, it is the energy currency of the cell. So moving on to this, now we know that this occurs only in the C3 plants, such as where the carbon dioxide compensation point is much more higher, like wheat, tomato, pisum, sativum, capsicum, and fascioles. That means beans. So in such plants, gossypium, cotton. So in such C3 plants where the carbon dioxide concentration is more, this photorespiration occurs. So what is photorespiration? The name itself says, if you break the word, we get photo and respiration. Photo or photon means light. So respiration is the presence of light that is occurring in the green cells is called as photorespiration. No doubt the respiration occurs during throughout the day and night, but in the daytime when the temperature is high, when the oxygen concentration is high, when the carbon dioxide concentration is low, that respiration that occurs is called as photorespiration. So what is the actual difference between normal respiration and photorespiration will be dealt at the last part of the video. So let us move to this photorespiration. So RUBP, as we all know, ribulose biphosphate, it combines with oxygen in the presence of low concentration of carbon dioxide. So the condition is the carbon dioxide concentration should be low and oxygen should be more. This RUBP combines with oxygen at that time and it forms phosphoglycolic acid and 3-phosphoglyceric acid. So the phosphoglycolic acid is a C2 compound. That means it has two carbon compounds. Hence this pathway is also called as C2 cycle or phosphoglycolic pathway. So the first step is the oxygenation of ribulose biphosphate leads to, in the presence of Rubisco enzyme, gives phosphoglycolic acid, which is a C2 carbon compound, and 3-phosphoglyceric acid. This 3-phosphoglyceric acid, it undergoes Calvin cycle to form the food product. Now what happens is this phosphoglycolic acid in the presence of an enzyme phosphoglycolate phosphatase. It gives off inorganic phosphate because there's only one phosphate group over here, phosphoglycolic. So it gives off the inorganic phosphate and it becomes glycolic acid. So this is the first step that is occurring in the chloroplast of the plant cells. Now this glycolic acid is transported to the peroxisome. There the glycolic acid in the presence of glycolate oxidase. So from the first video I am saying that to remember the enzymes you need two names are there in the enzymes. Like we have the scientific binomial nomenclature for plants. For enzymes we have the first name that indicates its substrate molecule. The second part indicates what function does that enzyme do. So here glycolate oxidase. So the substrate is glycolate or glycolic acid and the function that is doing is oxidation. So it oxidizes glycolic acid to form glycoxylic acid. So there is differentiation, there's just a minute difference. Glycolic, glycoxylic. So there's much difference. So glycolic acid undergoes oxidation in the presence of glycolate oxidase to form glycoxylic acid and hydrogen peroxide. This hydrogen peroxide is highly toxic to plants. So as and when hydrogen peroxide is formed, it catalyzes to form water and oxygen in the presence of an enzyme catalases. So H2O2 is formed along with glycoxylic acid. Since it is toxic, it is broken down into water and oxygen. Later, this glycoxylic acid is converted into glycine and alpha ketoglutarate. glycine and alpha ketoglutarate. Now this reactions occur in the peroxisomes. Peroxisomes. Now the glycine from the peroxisome moves on to the mitochondria and it combines with the water. So water is formed in every step almost. So the glycine combines with water to form serine. While during this process is occurring the presence of an enzyme glycine decarboxylase. Glycine, it indicates the substrate molecule. Decarboxylase, that means function is decarboxylation, removal of carbon dioxide. From where? From the glycine molecule, the carbon dioxide is liberated, along with that ammonia is also liberated to form serine. So you can see carbon dioxide and ammonia is liberated out, and NAD forms, reduced NADH2 is formed, which is an energy rich molecule. So serine is formed in the mitochondria. Now the serine that is formed in the mitochondria, it transfers to the peroxisome and then it converts into glyceric acid. So in between there are various intermediate complex steps. Those are not necessary. For those who are interested, can refer some other textbooks for those intermediate steps. But these are simple steps and very important steps that I made to make this topic very easy and simple. So the serine now is converted into glyceric acid in the peroxisomes. So the glyceric acid does not have any phosphate molecule. This glyceric acid directly it is transformed into chloroplast. In chloroplast, what happens is there is phosphate molecules formed because phosphoglycolic acid gets converted into glycolic acid by the removal of phosphate molecule. This is the first step. So you can find many inorganic phosphate molecules in the chloroplast. Utilizing all those inorganic phosphate molecules, the glyceric acid becomes phosphoglyceric acid. That phosphoglyceric acid, it readily enters into Calvin cycle and the food is formed or the carbohydrate is formed. The sucrose or glucose or sugar molecule is synthesized. So this is about the photorespiration. So this is very simple where the oxygenation of ReBP takes place, giving us to phosphoglycolic acid that becomes glycolic acid. Glycolic acid becomes glycoxylic acid. Glycoxylic acid in H2O2. H2O2 is highly toxic, so it's readily converted or broken down into water and oxygen. Glycoxylic acid is converted to glycine, glycine into serine, serine into glyceric acid into phosphoglyceric acid that enters Kelvin cycle. So this completes your photorespiration. Since the starting stable first product is a phosphoglycolic acid, which is a 2-carbon compound, this cycle is called as C2 cycle or phosphoglycolic pathway. Now, what are the factors that are involved in this entire photorespiration? So, there are many factors like the temperature should be very high, high temperature should be there, and light also should be there. So, that is the definition. This is light induced process and it occurs only in the green cells. And also, the oxygen concentration should be very high and the carbon dioxide concentration should be very low. Carbon dioxide compensation point should be very high. So these are all the factors that are responsible for this photorespiration to occur. And what is the significance? When this scientists discovered photorespiration, they thought it's usually a waste process. 75% of the energy is wasted by photorespiration. Later they came to know that, if you've seen my previous video in introduction to respiration, I mentioned about solarization, photooxidation, photoinhibition. So please do watch that video. What are those means? When the intensity of light increases, the chloroplast organelles and other cell organelles are broken down. They are denatured. So this photorespiration prevents all those things. It prevents the solarization. So it prevents the breaking up of the cellular elements or the chlorophyll elements. That is one of the major significance. And also in that introductory video to respiration itself, I mentioned factors responsible for respiration. So if the accumulation of oxygen takes place, the respiration level goes down. Accumulation of the end product. So if here also due to high oxygen concentration, the respiration is lower down. Since oxygen is utilized by this photorespiration process, such things and all is not happening over there. Usually when oxygen is accumulated, the respiration level goes down. So this prevents the accumulation of oxygen because oxygen is readily utilized by over here. This is another significance. Now coming to the last part, what is the difference between photorespiration and normal respiration? In normal respiration, if you see, the substrate molecule is carbohydrate, protein or fats. But here the substrate is phosphoglycolic acid. In normal respiration, it occurs 24 hours throughout the day and night. But here it occurs only in the presence of light, that is during daytime. And also in for respiration, it occurs in all cells. But here it occurs only in the green cell because chloroplast is necessary. So it occurs only in the green cell. And also one more important thing in respiration, what happens is it occurs in all the plants, all C3, C4, CAM, succulent, all types of plants it occurs. But in photorespiration, it occurs only in C3 plants where the carbon dioxide compensation point is very high. Only in such C3 plants this process occurs. And in respiration, you have the involvement of glycolysis, Krebs cycle, terminal oxidation, so many complex steps you have. Here you have only C2 cycle of phosphoglycolic pathway. It is very simple. And also in respiration, there is only involvement of two organelles, cytosol and mitochondria. Here there is involvement of three organelles, chloroplast, peroxisome and mitochondria. And in case of chloroplast, in normal respiration, no hydrogen peroxide, no ammonia is formed. Here you can see hydrogen peroxide, ammonia is liberated out. In normal respiration, where there is abundant ATP molecules are broken down, 38 molecules of ATP is formed, but here there is no ATP molecule synthesized at all. So this is the difference between respiration and photorespiration. And also this photorespiration is dependent on temperature, dependent upon light, dependent upon oxygen carbon dioxide ratio, whereas the normal respiration, it is independent of all these factors like temperature, light, though they are independent to a certain extent, but not so much as this much is independent. So that is independent of temperature, light, oxygen carbon dioxide ratio. So this is a very important for it is for green plant, C3 plant that is growing in high light like equatorial region or in desert region and all, where some C3 plants are growing where that light illumination is very high and temperature is very high. This is a very necessary step to prevent solarization. So I hope this video was very useful for you and I made it simple. Thank you for watching. Please do like and subscribe to my channel.